Now, let's see here. The restriction enzymes are actually not, they're not just chopping, okay? They're actually going to cut, they're going to de-bond, basically. They're going to cut the covalent phosphodiester bond between two nucleotides. Okay, that would be the sugar phosphate, okay? They do it in two ways, okay? There's one that's really common, which is called the staggered way. All right, that means that you have your double-stranded DNA. Okay, now the restriction enzyme will recognize this spot right here. Okay, this right here. So it'll cut here and it'll cut there. All right, on each side. It recognizes the little pin tips here. All right, and that's where it'll cut. So it'll cut like this and then it'll cut here. And that means that when it's cut now, you've got these sort of staggered ends, okay, where the DNA is going to be double-stranded at some point, and then you'll have a hanging piece that is single-stranded. Okay, that's called a staggered cut. All right, and they call the end of these cuts sticky ends because you have an overhang of single-stranded DNA. And these are good for when you are trying to recombine two pieces of DNA, because now that single-stranded end will complementary pair with another single-stranded end of another sticky end of another plasmid. Okay, so the less common way, okay, would be if you have your two single-stranded pieces of DNA. Okay, they recognize this site right here and they chop right there, down that. So now when they chop, you've got two blunt ends. Okay, which means that there is no overhang. It's all double-stranded the whole way. These are less common because now it's going to be harder for you to complementary base pair anything right here. Okay. You like my little, let's see, that's improv. Okay. All right. So now that we've got our recombinant DNA, all right, you've got your little sticky ends. They're going to complementary base pair. All right, with temporarily by hydrogen bonds. Now, hydrogen bonds, remember, are like the weakest bonds that you can have, so it's only temporary. So then you would add DNA ligase, which will actually go back and rebond they'll build the backbone part so that the recombinant DNA is actually stuck to each other. Okay, so now you have your recombinant DNA. Now, how do you clone it? All right, so remember that one end of our plasmid is going to complementary base pair with the end of our gene of interest, okay? And that's how we're going to insert it into our whole big plasmid on each side. So now we have our vector, okay? And a vector is going to be anything that can carry and copy our DNA, okay? So our bacterial plasmid is called our vector now, our cloning vector. They're easy to isolate. It's easy to reintroduce the vector back into the bacterial cell, and then it's really easy for it to replicate because it just replicates along with all the other cells as it replicates, okay? Now, we're going to go over the steps in detail. I'm going to go through it kind of fast because I think this is not really that hard to get, but you can always email me or call or text me if you have questions about this, okay? Okay. So, in the book, on figure 20.3, okay, they give you an example of how they would screen for a specific vector, okay, or a specific recombinant piece of DNA. Now, we've already isolated the vector, we've already isolated the gene source DNA, which is our gene of interest, and now we're going to put them together. Okay, so quickly, your plasmid has to have a marker on it. Otherwise, how are you going to be able to tell whether or not it was there? Okay, whether or not the recombinant DNA is. So what I mean by marker is, in the book, they give you an example. Your plasmid has an AMPR gene, which is the gene that helps you become resistant to ampicillin, the antibiotic ampicillin. And then it also has a LAC-Z gene. okay, which is the gene that codes to break down beta-galactosidase or something, okay, or beta-galactose or something. Okay, so, and this LAC-Z gene is where the restriction site is. Okay, so this is where your restriction enzyme is going to recognize and cut, right in between the LAC-Z gene. Okay, so you put your plasmid into a solution. You put your DNA of your gene of interest, or your DNA that has your gene of interest into it. You put that in the solution. You put a restriction enzyme in there that's going to recognize the restriction site at the LAC-Z gene, within the LAC-Z gene. All right, it digests the plasmid, it digests the DNA. And that way, you always want to use the same restriction enzyme, because then it's guaranteed to recognize the sequence on both the plasmid and on your gene of interest, so that way when they complementary base pair, it'll be easier for them to hook up and become recombinant DNA. Okay, so your restriction enzyme is going to cut at the LAC-Z gene, right in the middle, okay, disrupting the gene. Okay, this means that now the coding sequence is off. It's not going to be able to make the protein or the enzyme that is going to be needed to break down beta-galactose. Okay, so the fragments get mixed together. They temporarily bond by hydrogen bonds, and then you add DNA ligase to ligate them together. Okay, now you have your DNA, your recombinant DNA. Now here's the caveat to this. Okay, you don't know how many restriction enzyme sites there are going to be on your piece of DNA. You know where your gene is, and you know that the restriction enzyme is going to cut your gene, but it's also going to cut many other pieces, all right, which also could be genes. You don't know. And all of those sites, since they're all complementary because they all cut at the same spot and they all recognize the same sequence, are going to be able to base pair with your plasmid. Okay, so now you have many different types of recombinants. This is where the screening later on comes in. So now you're going to have many recombinant DNA pieces. You don't know which one is your gene, all right, but later on we'll be able to figure it out, okay? Okay, so then you got to reintroduce it back into the bacterial plasmid. Back into the bacterial cell. Okay, the bacteria are going to uptake this plasmid by transformation. Okay, remember that transformation is the uptake of naked DNA. Generally, it's a plasmid. Okay, it's usually, generally it's a DNA that they lack. Okay, so if I have a LAC-Z gene plasmid, okay, with ampicillin resistance on it, then I need to put this plasmid into a bacterial cell that that does not have the LAC-Z gene. Okay, so it would be LAC-Z minus, right? Because it has a better chance of uptaking that DNA because now it doesn't have the ability to make the enzyme beta-galactosidase, okay, which is the enzyme that's going to cut up the galactose. It doesn't have that, but the plasmid does, so it's more likely to uptake that and be useful in some way than if I did it in a LAC-Z plus bacterial cell that already had the LAC-Z gene, okay? Because now, if I have a LAC-Z gene plus bacterial cell and I try to put this LAC-Z gene that's interrupted into the cell, when I go to screen for it, it will look like it could have the plasmid. It could have my recombinant DNA plasmid, but it would still be making LAC-Z. Okay, do you understand? Because now it still has the LAC-Z protein, and you've given it a plasmid that is interrupted. It doesn't care that it's interrupted because it already still makes a LAC-Z. So it'd be easier to see it in a LAC-Z minus. Okay, there are other ways to screen for it, but that's the easiest one to do. Okay, so then you clone the cells. Now you're going to plate them out on some kind of agar medium. Okay, which has the nutrients and all that other junk that it needs. And then your colonies start to grow. Okay. You leave it overnight in a specific temperature and it'll grow. Okay, you make sure that on your agar you have ampicillin antibiotic in the agar. All right, because then when you're screening, all the colonies that grew have the ampicillin resistance gene. Okay, which means they have the bacterial plasmid. Not necessarily your recombinant gene of interest, but they at least have the plasmid. Okay, so now you know that all of those that are plated onto the plate that grow have the AMP-R gene, okay, plasmid. Furthermore, all right, if it has an interrupted LAC-Z gene, right, then it won't make the enzyme it needs to break down galactose, which you've also put into the medium. All right, and when it breaks down galactose, it turns like a blue color. So if it has an intact LAC-Z gene, it makes beta-galactosidase, it breaks down galactose, the colony turns blue. Okay, you don't have that, okay, because your recombinant DNA has disrupted it somehow. So now you know that the white colonies, the ones that didn't break down beta-galactose, don't have beta-galactosidase because the LAC-Z gene isn't intact and it has some sort of recombinant DNA, but you're not sure which one yet. So those are the ones that you want to focus on. Okay, so in the book it has like 5 white colonies on the little picture on figure 20.3. One of those 5 could have your restriction enzyme in it. I mean your recombinant DNA that you are interested in. But you don't know which one yet. But you do know that they do have the plasmid. Okay, so that's one part of the screening process. Okay, now, how are you going to figure out exactly which one of these recombinant DNAs is the one that you want, that you're looking for? Okay, they use a method called nucleic acid hybridization. Okay, and they use a probe basically, and what they do is you have like at least some sequence part of your DNA that you're interested in. Okay, you've got at least 15 base pairs of it figured out, the sequence. And so you make a primer, basically. You make a short, single-stranded piece of DNA that is radioactive, which means that you've used fluorescent nitrogenous bases to put it together. And it will complementary base pair with your gene of interest. Right? Okay, you following me? And so then you can run it on a gel, and whatever part flashes fluorescent, you know, okay, that is the bacteria that's got my specific recombinant DNA gene that I'm looking for. Understand? Okay, so now you've figured out which recombinant DNA is yours, the one that you want. Now you grow that up. You grow it up, you grow it up, you grow it up, and then you isolate it. And then voila, you've got your little replicated recombinant DNA. Okay? Now, now that we've got it actually replicated, how do we get it to express? Okay, because you've put it into a prokaryote. Eukaryotic genes don't express very well in prokaryotes because prokaryotes don't have the mRNA machinery, the RNA splicing machinery that you need. Okay, there's a lot of incompatibility going on here. So how are you supposed to get it to express? All right, now, scientists have also figured that one out. They've come up with something called an expression vector. Okay, it's a cloning vector that has already a built-in promoter for your restriction sites. Okay, so it's a promoter that's upstream of the restriction site of your gene, and that way your bacterial cell doesn't have a problem finding whatever gene you want replicated. Whatever gene you want transcribed and translated, because you've built in a promoter already for it to recognize. Okay, but now, so now it can recognize it, it can transcribe it, but remember that bacterial cells, they don't know what introns are. They don't have them. All they have is the DNA, and the whole thing is transcribed, it's translated simultaneously, and then it goes. Right? They don't do any of the splicing junk that eukaryotes do. So now what? Bacteria lack the RNA splicing machinery for introns. So scientists came up with another thing. All right, we'll make an artificial eukaryotic gene without any introns in it. And what do? How would you make a eukaryotic gene that doesn't have any introns in it anymore? You would use mRNA, right? The RNA that's already been spliced and it's already got its 5' cap and it's already got its poly A tail. You use that mRNA, right? So you extract the mRNA from the cell and use that to make your DNA. mRNA to DNA. Now what do you have to have? You have to have reverse transcriptase, right? So you take out your mRNA that's already got all its introns taken out, it's already got all of its modifications done. You use reverse transcriptase to make the DNA complement to that. So it's called cDNA because it's complementary DNA. And then you put that into the vector. Okay, so they found ways around all of this incompatibility junk, right?